we will describe a little bit about the molecular characteristics of the contractile filaments. So first thing we need to know is that our muscles have smaller units. So it's muscle, fascicle, muscle fiber, myofibrils, which are also known as sarcomeres. And there's two components that are very important here. It's actin and myosin. And they have different components. So the end lines are called Z lines or Z discs. And then we have our actin and our myosin in the middle. So when we have both that are overlapping, myosin and actin, that's called the dark or the A band. So that's thicker. And when we just have actin, it's called the light band, which is thinner, or it's also called the I band. So let's look at how the sarcomeres come into play when we are contracting and relaxing our muscles. So there's something called the neuromuscular junction. And you basically have an action potential coming from your brain to your neurons, to your sarcomeres. And you have your snare proteins that are binding the vesicles, which have neurotransmitters. In this case, it's ACH or acetylcholine. And when your action potential comes, it lets the voltage, calcium, and sodium channels come in, which will bind to your vesicles, which will leave your ACH out of the cell. So we're in the ECF right now. Then it binds to your ionotropic receptor, in this case it's N1, and it's ligand gated. It will let your sodium in, and that will create a sphere of influence. And that will open another channel. And when this channel opens, it comes into something called the DHB. So we have two very important things here. You have your RYR, which is also known as the ryanodine receptor, and your DHB. DHB is very sensitive to calcium. So when we have a positive ion come in, the DHB will open, and since it's bound to RYR, it will open as well. IYR is directly attached to your SR, and your SR has a lot of calcium. So it will let the calcium out, and whenever you have calcium in your muscle, it will help contract. So we will talk a little bit more about what happens when you get calcium in your muscle or your sarcomere. So normally, you have kind of like this chain, and all of this is actin, but let me explain to you how it works. So you have kind of like your monomers, those are your G-actin. G-actin have also binding sites on it, but when you have a ton of G-actin together, that's called an F-actin. Around it, you have something called tropomyosin, and you have your myosin heads trying to attach to your binding sites, but your tropomyosin is in the way, so you can't contract. And you have something else, looks kind of like a Mickey Mouse here. It's called tropomyosin. So you have three parts, tropomyosin T, I, and C. C attaches to calcium, so you have 4 calciums in order for it to be activated. You have 2 of high affinity and 2 of low affinity. You have your tropomyosin, that's directly to your troponin T, and you have your D-actin, which likes to attach to your actin, your G-actin or your F-actin. So when your muscle is relaxed, you will be in a high energy conformation or cocked conformation. It would kind of like look like this. And it's not attached, your myosin is not attached to your actin. You have your tropomyosin in the way. However, we already have our calcium in our sarcoplasm from our sarcoplasmic reticulum that came out. So because it bound to troponin C, it moves tropomyosin out of the way. And so it lets myosin go into the binding sites. So what will happen? You will release heat, and then you will contract. You will literally have a power stroke that will literally like push away your phosphate and your ADP. And as you can see, our angle is a lot less. So we call this the on-cocked or our low energy. So literally, this is what happens when you go to the gym. Like you're like this and you make a curl, and you're in your on-cocked or low energy. Once you're there, you can't really have the same strength. So you need to come back to relaxation in order to do another curl. So you come in and you bring an ATP, and that releases heat. and brings it back into the conformation you had before. But how do you do that? You need to hydrolyze that ATP in order to have ADP and phosphate again. How do we do this? Let's come back into the neuromuscular junction. So you have a lot of calcium outside, your muscle is contracted, and so you need to bring that calcium back into this SR, the sarcoplasmic reticulum. However, your concentration in the sarcoplasm is a lot less than the concentration of calcium you have in, so you need an active transport. We will do that using ATP through the sarcopump. It takes longer, and it's going against the gradient. So that's how you do it. You transform ATP, you hydrolyze it. ATP literally like breaks down into ADP and phosphate. Okay, let's talk a little bit about what energies we use and how the muscles work. So you have aerobic and anaerobic, and you have slow and fast. So we will break it down a lot from here. So let's come back here. Where do you get your energy? You have anaerobic and aerobic. So when you don't have oxygen, the first thing you do, you get ATP from your cytosol. And that lasts between 1 and 5 seconds because it's literally there. You have it readily available for you. The second option is to use your creatine phosphate. It lasts up to like 10 seconds approximately. And you use an enzyme called creatine kinase. Your third option, it lasts longer, but you will need to break glucose. So it's called glycolysis, lysis for breaking down. So you can have 2 ATP per 1 glucose. And the most reliable, but the one that takes longer, you will need oxygen for that. Aerobic respiration, it lasts for hours, but you will need to use your mitochondria for that. So let's go through this chart and let's talk about like the different muscles that you have and how they work. I'm going to here. Okay, so first you have your slow. You use your myosin ATPase 1. It's very small. It's extensive in blood supply, so that's why it's red. You have a low power profile and you don't have fatigue. And an example of this is just like walking. You don't need a lot of energy, but you can do that for a long time. So you use oxidative energy resource. There's another aerobic that's a little bit faster. You use 2A myosin ATPase rate. It's medium size, medium blood supply, it's still red. You have a high power profile. You do get fatigued a little bit here, it's medium. And then your energy source is still oxidative. The last one, anaerobic. Your myosin ATPase is 2X. It's very large and your blood supply is small, so it's usually white. And you have a very high profile. So you can do like very high intensity cardio here. However, your fatigue is very fast. And you use glycolytic energy source. So this is why when you do like a lot of Tabata workout, for example, you do a lot of high intensity training and then you rest for a little bit so that you can come back to whatever you're doing. We're talking about fatigue here. So when we think about fatigue, it's not that you don't have energy or that you're lacking ATP. Actually, you can feel fatigue when you still have 70% of your ATP in your muscle. So it's generally because you have an ion imbalance. We have 2 different types of fatigue. We have psychological from your central and you have physical from your peripheral. We won't talk about psychological in this case, but physical does have a low frequency and a high frequency fatigue. So whenever you have low frequency, it's because there's something going on with your calcium, so it takes longer to recover. There's an imbalance in your ryanodin, and it's inhibited by magnesium. There's something with your sarcopump, or you need to give time for your sarcopump to help balance that calcium back into the sarcoplasmic reticulum. So a good example is like long hikes. You can do that for long periods of time. You can hike for hours. However, you'll take probably a couple of days to recover. And then you have your high frequency. So it's usually because of a membrane imbalance. So in order to help you here, and we do have a fast recovery, we use our sodium potassium pump, chloride ions, because there's like a lot of positive things inside the cell. So if you bring chlorine, it will help balance the charge. And so you help with your membrane potentials. There's an impaired membrane excitability, which means that your ability to reach threshold is impaired. There's a potassium buildup in the T-tubules. So as we learned in class, T-tubules is basically when your membranes are not like flat. You create basically a tube, and so there's a lot of concentration of your positive charges from your potassium. But if you give it like 10 seconds, it will diffuse and you will be able to come back to your activity. Thank you. Okay, now we're going to talk about the smooth muscle and how it contrasts to the skeletal muscle. So a general cycle that we talked previously is that you just have your tropomyosin, you have your calcium come in, it gets rid of that, or, well, it doesn't get rid of it, but it just puts it aside so that you have space for your myosin to bind to your binding sites in your D-actin. So really briefly, we have our cocked high energy, we put the calcium in, you release heat, you uncock, and you have your power stroke. So although you have your angle come a little bit less here, the power stroke will make literally your muscle contract into your uncocked low energy. You have your ATP come in, then you hydrolyze it through your sarcopump, and you come back to your cocked or high energy. So let's see how it works with our smooth muscle. So in our smooth muscle, we basically have two types, tonic and phasic. So we know that our smooth muscles are a lot of sphincters. So we have sustained contractions, and we also have rhythmic contractions like peristalsis in your digestive system. Let's talk about tonic. I will explain a little bit about rho kinase and what it does, because we will use it both in the tonic and in the phasic, but I'll explain it more in depth right here. So rho kinase, its function is to phosphorylate MLCP through our phosphatase. So let's say this Pac-Man is our MLCP and it's active, and so it's literally thinking, okay, I need phosphate. And so rho kinase says, no problem, I'll give you a phosphate, and then it inactivates our MLCP that was active before. So you have your myosin, it's not bound yet, and you have your ATP and your phosphate. And then a lot of things happen. Let me explain. So you have your calmodulin here. It's inactive right now, and as you can see, it has 4 calcium binding sites. So once you bring calcium in and it's inactive, you have 4 calciums bound and it changes its conformation. And then it travels to your MLCK, which is inactive right now, as we can see. It's basically like as if it has something in its mouth, like braces or something. And so once you have your active calmodulin trying to come into your MLCK that's inactive, it changes its conformation. So MLCK, when it's activated, it kind of looks like this. So you basically take this thing, its tail, out of its mouth, and you also have your calmodulin here. So how does this come into play? When we come from the first to the second step, we bring out calcium, our ATP, and our MLCK with phosphate. This phosphorylation causes it to bind to actin. And right now, this phosphorylation will come into play instead of the troponin C that came in between the first and the second step in our skeletal muscle. Remember that troponin C was the one that bound to the tropomyosin and gave it space so that our myosin would bind to our binding site in our G-actin. So we don't have that in skeletal muscle. That's why we need the phosphorylation in MLCK to bind to the actin. That's the main difference, and something very important to notice. Okay, so once you do all of this, you have an extra phosphate. You have one phosphate here, ADP. You have your phosphate still, your ADP, and one more phosphate. Our phosphorylation again comes into place instead of troponin C. And then we have our power stroke. You get rid of your ADP, of your phosphate, MLCP comes, and then your phosphate that we brought in between the first and the second step goes out. So now we have our uncocked or low energy in our myosin. You bring ATP, you bring it, you bring that angle back again, and then you hydrolyze your ATP because we want it to be ATP and phosphate. Again, hydrolyzation is breaking down your ATP because T stands for tri and D stands for di, so that's why you have an extra phosphate in there. And again, in order to bring calcium back into the SR, or the ECF in this case, you use your circuit pump. So tonic favors calcium from the ECF. You basically have both, but you like ECF better here because it's sustained, so you need a constant source of calcium. Okay, phasic is different because these are rhythmic contractions, so you don't need sustained calcium sources. So it favors calcium from the SR in the ICF, or the sarcoplasmic reticulum. It's pulsing and it's very fast. So we will talk about rhokinase and calmodulin again, just as all of these steps, but I won't explain it again. So you have your ADP, you have your calcium, ATP, MLCK, and phosphate come in. You have the same second step as before, and now you have a power stroke. Your ADP and your phosphate get out, and notice that we are in our uncocked or low energy position, but because we don't have MLCP come in, we don't get rid of the phosphate. So we still have this phosphate right here. You have your ATP come in, and it's still there with your phosphate. So there are two things that can happen in here. You can basically jump from your fourth to your second step, or you can come back. So you can just bypass the first step and come back into having our ADP and 2 phosphates, or you can hydrolyze and bring calcium to where it came from through the circuit pump again, and then you just repeat that cycle. Okay, MLCK means more phosphate, MLCP means less phosphate. If you have more MLCK than MLCP, it's likely to be phasic. If you have less MLCK than you have MLCP, it's likely to be tonic. Smooth muscle has high chain regulation. So I would like to compare a little bit between the skeletal and the smooth. So smooth is autonomic. You use calmodulin, MLCK and MLCP, and you don't have T tubules. In the skeletal, it's regulated by the somatic nervous system. You have troponin and T tubules. And just a little bit more to notice is that skeletal is somatic. You use it for locomotion, it's voluntary, it's rapid and forceful, and it's usually quick fatigue. And smooth, it's involuntary. You have no fatigue. It would be very bad if you would get fatigued in this autonomic things that you can't control. And it's low sustained, depending on whether it's tonic or phasic. A little bit more about the structure. I would just really touch it a little bit because number 3 talks a little bit more about this. But in the smooth muscle, it's spindles happens in the cells, it's single nucleus. And then in the skeletal one, it's striated, and you normally have multinucleated things. And they're attached to bones and tendons. Oh, I forgot something. Let's talk a little bit about what happens in the enzyme activation between phasic and tonic. This is basically what I just explained, just in a little bit more concise way. But let's say that the red is calcium, the blue is MLCP, and the black is MLCK. You can notice that in the phasic, our MLCP and our MLCK have a big gap. However, in the end, our MLCK and our MLCP have a lower gap between them. Yeah, thanks for watching. Here are a little bit more comparisons between skeletal and smooth muscle. So we always have ryanodine, DHP, T tubules, tropomyosin, and we never have MLKC or MLCP for skeletal muscles. For smooth muscles, we don't need action potentials, they're independent. However, in skeletal muscles, we always need them. Smooth muscles can be activated through hormones. We also have ryanodine, but we also have IP3. We don't have DHP because they're only attached to T tubules, and we never have T tubules in smooth muscle. We have no tropomyosin, And instead of that, we have MLCK and MLCP, and it can be tonic or phasic. Okay, just to recap a little bit on smooth muscle, this is how a typical smooth muscle cell looks. We have G proteins that work as a cascade, and whatever subunit they release goes to phospholipase C, which release IP3, and that will cause calcium from the SR to go out of the cell. And for that to come back into the cell, we will use an activated pump called the Circa pump. We also use antiporters. For example, we take sodium out of the cell and calcium inside. Sometimes we need to bring things against its gradient. We have calcium ligand gated channels. We have calcium channels that are also stretchy or mechanically gated. We have DHP, that's very important in order to get calcium inside of the cell. And those are some of the channels that are important for our smooth muscle cells. Okay, we will start talking about the muscle receptors. So there's two main ones that we want to know for proprioception. Those are the Golgi tendon organs and the muscle spindle. So come here with me and we will learn some features about it. So first, the muscle spindles are located within the muscle belly, while the Golgi tendons are within the tendons. The stimulus for each of them for the muscle spindle is the muscle length and the rate of change, but for the Golgi ones are for muscle tension. The sensory fibers for muscle spindles are group 1A and 2. Golgi tendon organs are group 1B. The reflex for muscle spindle is stretch reflex or myostatic reflex, but the Golgi tendon organs are the tendon reflex or the inverse stretch reflex. This one's a very important one, specifically when you're very contracted. A very cool reflex that it does is that it lets you stretch farther the more you contract, and we will talk a little bit more in depth about this later. The effect of the muscle spindle is that it contracts stretched muscles, and the Golgi tendon relaxes contracting muscles. So it's exactly the opposite in here, but they work together to ensure right proprioception and to avoid lesions on our muscles. So together, they regulate muscle length, they prevent overstretching, monitor muscle tension, prevent excessive force, provide essential sensory information for proprioception, which is the body's awareness and ability to know its position and movement in space. And together, they ensure smooth, coordinated, and safe movement by regulating muscle tension. Okay, so just as we talked about before, Golgi tendons are specifically in sensing muscle tension. Muscle spindles are very aware of muscle length and producing muscle contraction. So something super cool that you can do is that if somebody doesn't know that you're going to like touch them or something, they would probably like try and shy away from you, but they will do it in a way that they won't fall. So it's very funny because if you touch somebody here, they'll do something like this so that they won't fall. And it's kind of cool to see that. So we will talk a little bit about some reflexes that we do. So we have the stretch reflex and the Golgi tendon organs. So what they do is that in their motor and neuron sensory, in the motor and sensory neurons, they are activated by the spinal cord. A good example of this is that if you are laying down and put your legs up and you contract a lot, then you can stretch a little bit more, and then you contract more and you stretch farther and farther and farther, and that's because of the Golgi tendon organs. The stretch are monitored by spindle. They're agonists of EPSP and they're antagonists of IPSP. The Golgi tendon organs are for contraction, and they're exactly the opposite as the stretch ones. They're agonists for IPSP and antagonists for EPSP. The withdrawal reflex brings part of the body closer to the center. So it activates nociceptors, the EPSPs to the flexors and the IPSPs to the extensors. We have the cross extensor reflex, and that responds to withdrawal reflex, giving the opposite response contralaterally. That means on the other limb. It produces the exact opposite thing as you're doing on the other side. We can see here a little bit about how our Golgi tendon organs look. We have a nuclear bag fiber in the middle and a nuclear chain fiber here that is there. And for the muscle spindles, we have, so this is the brain, spinal cord, and then your muscle fibers. And you have alpha motor neurons, you have the gamma motor neurons, and then the sensory ones. So this is your muscle spindle apparatus. And we also talked a little bit about static gamma motor and dynamic gamma motor. So this is how they work. The static one uses the second, and the dynamic uses 1A. Okay, let's talk about taste. So we basically can taste 5 different things: salt, sweet, bitter, umami, and sour. And spicy is actually a pain receptor or nociceptor channels, and that's modulated by capsaicin. However, we will talk about the other 5 right now. Although we have 5 different ones, we have 2 that are ionotropic and then 1 that is GPCR or G protein receptors. And we will join the sweet, the bitter, and umami here. So for salt, we have an ionotropic receptor. So the ion that we will be using here is sodium. That's an epithelial sodium channel. That's a leaky channel for sodium. And we already know that a lot of positive charges depolarizes the cell, creates an action potential. And although the action potential is not in that cell, the acetylcholine that we release here will bind to the next cell, and then the action potential will be in the next cell. That's very important to know. We don't do that action potential in the cell that we're receiving the ions. Okay, sour. We know that the more protons we have, the more acidic something is. We still have the epithelial sodium channel, but we will have protons come in. We will have potassium leak channels, and then calcium comes in. So our protons come and depolarize our cells. Okay, let's talk about the sweet, bitter, and umami. These are G protein receptors, so it won't directly come in, but sweet are carbohydrates, quinones are for bitter, and glutamate are for umami. So those will basically be the things that will bind to our G protein. Once that is activated, it will activate something else, and then we will have IP3 come in to our SR. And then our SR will release calcium, and then that calcium will help release our neurotransmitter from blood vesicle. Something very important to note here is that for the ionotropic, we're bringing things from outside the cell to depolarize it. However, for the G protein in here, sweet, bitter, and umami, we're using calcium inside to depolarize the cell and cause the oxygen potential. Let's talk about smell. So in taste, we talked about IP3 being one of the main important things. And here is cyclic AMP. So let's say this is a nose, and then on top of your nose, we have a cribriform plate. And there is something very important by the cribriform plate. It's an enzyme called adenylate cyclase. So the chemicals or the smelly stuff that come in are basically the things that will act as your neurotransmitters and then go straight to the neuron. And neurons normally don't regenerate, but this one's do. So I think that's cool. So you have your adenylate cyclase come in, you have a G protein here. So if your autoron comes here, it goes and creates cyclic AMP, which will open a channel that lets your sodium and your calcium come in. And then you have chlorine come outside. And that's how we smell. So CAMP is released by adenylate cyclase and then opens these channels. And then our voltage gated chlorine one opens, and both of these channels help depolarize the cell to create the action potential. Let's talk about hearing. This one's a very cool one too. So I will need to explain several things before we know how it actually works. So we have a conversion of sound waves through action potential. So there's something called an inner and outer hair cells, and this is how they work. We have this inner hair cells, we have potassium, it comes in, depolarizes the cell, we let calcium in, and then we release neurotransmitters, just as we've learned throughout the class. So the outer hair cells amplify the movement, receive something and they amplify them. The inner hair cells are the ones responsible to send the action potential. And then the tension pulls ion-gated channels. Okay, we'll later talk about something called the stria vascularis, and that will be responsible for potassium driving force. It will be responsible to actively secrete potassium from the perilymph fluid to the endolymph fluid. And it will do this through the sodium-potassium pump and through something that we call the NKCC channel. It brings sodium, potassium, and 2 chlorines inside of the cell. And then sometimes we see something exactly the opposite as the sodium-potassium pump, because we will put 3 potassiums out and 2 sodiums in. So it's completely backwards as the sodium-potassium pump that we have learned so far. Okay, in order to perceive sound, there's something called amplitude and frequency that we need to be aware of. The greater the amplitude, the greater the frequency, and more action potential we will have. Okay, and the frequency of the wavelength determines which portion of their auditory cortex receives the action potential, because we have different parts in our inner ear that are sensitive to different wavelengths and frequencies. Sometimes we will want to inactivate hair cells, and although we normally think as needing to hear different things, we sometimes need to focus on other things. For example, when you're sleeping, if we inactivate our hair cells, we will dampen the signals and we'll be able to have a better sleep. Or sometimes we will just want to focus on specific sounds. And so if we dampen other signals, we'll be able to focus on that one sound that we want to hear. For fine-tuned hearing, our brain sends efferent signals that use acetylcholine to open non-selective cation channels. And this will cause calcium to flow and open the potassium channels, and that will hyperpolarize our cell. And then we can send that from the auditory cortex to other hair cells. Okay, let's talk about what happens in our inner ear and the parts. We have the endolymph and the perilymphs. So we already talked about how the perilymph sends fluid into the endolymph, which is more inside. So we have osmolarity of 150 in our ECF, and we don't really have a concentration gradient here, but there's something very important to know. Across our chambers, our resting membrane potential is 80 millivolts. Inside of the cell is -40. So the different concentrations of osmolarity and the different charges that we have will influence a lot in how our ear works. So we have 120 in the endocochlear potential from the perilymph, but as we said before, we just have an 80 millivolt resting membrane potential in the endo part of the cell. Across chambers and inside we have a -40. So let's see what happens. We talked about this stria vascularis before, and this is just a depiction of how it works. We have our sodium-potassium pumps and then our NKCC channels here, and we can see those ions coming in. We still have leak potassium channels to keep the osmolarity. This is our outer hair cell, and these are our inner hair cells. So we have a lot of potassium leak channels here to help us keep the charges and the resting membrane potentials for the different parts of the cell. We need to remember that the outer part of our ear hair cells amplify the sounds. And we will talk about this part right here, the inner one. So when our muscles or our tendons or parts of our skin, in this case our inner ear, is stretched, then we will open mechanically gated channels. The only way we can open those is literally through tension. And that tension is created by the sound waves we perceive. So in this case, we will have potassium coming in, and then we will have leak channels as well. The reason why this helps us is because it lets calcium in, and that's how you create an action potential into the next cell. So outside, it's a positive charge, and inside, in our ICF, we have a -1. And this is the peri side of the lymph, because we have fluids in the perilymph and the endolymph, but the different pressures are the ones that help us create the sounds. In the perilymph, you have a concentration of about 7 millimolar of potassium, while in here you have like 150 milliosmoles. So that's a great concentration difference in there. And then you also have a very high concentration of sodium. It's about like 140 milliosmoles. These are our cochlear duct. We have our ear canal here. We have some very important small bones here, the malleus, incus, and stapes or something like that. And we have our cochlea here. So we already talked about the perilymph. So it's on both sides around, peri is around, and then endo is inside. That's the endolymph that we talked about just briefly before. And this is our basilar membrane. Our basilar membrane has different parts that are sensitive to different sound waves, as we mentioned before. Yeah. This is question 4, and it asks how the eye focuses on the light, the role of the ciliary body and the suspensory ligaments surrounding the lens. 2 types of photoreceptors. We will explain how they convert light into electrical energy and how center and surround cooperate in visual fields. So for this, we'll take a look at the eye, and it's a cross-section of our eye. So we have our cornea here, which is responsible of refracting light. More inside, we have our lens, which focuses our light, and surrounding our lens, we have suspensory ligaments and ciliary muscles. So our ciliary muscles work with our suspensory ligaments. So they contract or relax, and that would have an effect on our lens. So let's say that this is our lens right here. And so our ciliary muscles and our suspensory ligaments are kind of like sphincters. So when they contract, they bring everything tighter, and that will make our lens get thicker. When it relaxes, it would make our lens be thinner. We will talk more about this later. Inside, we have a fluid. It's called vitreous humor, and it's responsible for maintaining the pressure in our eye. In the back side, that's when our light converges, hopefully, and it's interpreted as an image that goes into our brain. So in here we have our optic disc. It's basically our blind spot because that's where all of our nerves are. Some other important parts are the fovea, which is where most of our rods are, and our macula, it's when there's our central vision. So we'll talk a little bit more about that. So let's talk about seeing things far and near. When we see something that's far, like me looking at the door, which is very far, my ciliary muscles and my suspensory ligaments will relax, and that will make my lens get thinner. And that's because I need to bend the light that's coming into my eye less. However, if something is near, like me looking at this whiteboard, my muscles will need to contract so that my lens will get thicker. And we might think, okay, why do we need this? Isn't it easier to see something that's super close? The thing is that I need to bend light more. So let's talk about some things that happen. For example, I have myopia. Some people have hyperopia. So let's see what's the problem and what happens in the eye. Hyperopia people are usually people that see better far than what they see near. So their problem is that their lens is too thin. So let's say that they're looking at a juice, and normally what it's in green would happen. So the light will come in, get into their lens, and their lens will converge the light, but it will converge it way later than when it touches the retina. So this is a retina right here. And so that's why they can't see very well something that's close. And we will need to correct that using a convex lens. Convex lenses, I tried to draw it here. It's this one right here. They're thick lenses and they bend light more. And they concentrate light more since it's thicker. So we have our image, and then what it will do is that it will concentrate the light right in the retina, which is before what they would do without glasses or without correction. Now myopia. Like me, we see very well nearsighted but not very well farsighted. So our problem is our lens is too big or too thick. So when we have light coming in from an object, it will converge way before our retina. So our image would be right here. However, our retina or our perceived image would be right here. And as you can see, it's bigger. It's not like it's not the point of the image. So we perceive that as a blurry image from things that are far. For this, we will use a concave lens for correction. Concave lenses bend light less and they spread the light. So when we have this, it will take longer for it to converge instead of converging way before our retina. So when we use glasses, we'll have our image right there in our retina and we'll be happy because we'll see well. Let's talk about some photoreceptors. So we have two different types. We have rods and cones. So our rods perceive white and black vision. They help us a lot at night. They're very sensitive to light, but they're not very high definition. Our cones perceive color. And our main cone colors are blue, green, and red. Everything else is just a combination of those three colors. Pretty fascinating. These are not sensitive to light. These have less pixels, but we have a better vision and more clear vision because they have a more precise intervention. In our fovea, we have a very high concentration of cones. Everywhere else in our eye is a combination of rods and cones. So let's talk a little bit more about how our eye converts light into electrical energy. This process is called phototransduction, and there are three basic things that we need to talk about: photoreceptor, bipolar cell, and ganglia cell. So our photoreceptor has different components that are very important. We have a protein here, and we have 11 cis-retinal. Let's say there's light. So we'll talk about light, what happens with light first. Our cis-retinal, when it perceives light, it changes in conformation into 11 trans-retinal. When it changes in conformation, we know that these proteins change their conformation. It's kind of like a cascade. So it lets an alpha subunit away. This will activate something called phosphodiesterase, or PDE. And what phosphodiesterase does is that it blocks something called cyclic GMP. What cyclic GMP does is that it's in the cytosol and it keeps our sodium channel opens. So what will happen if we activate all of this? So it changes from cis to transretinal, it gives its alpha subunit away, it activates PDE, and then it blocks our cGMP. So we don't have cGMP anymore. That means that our sodium channel is closed. Because our sodium channel is closed, our calcium channel will be closed as well. So we won't be able to depolarize our cell as much. So we won't have glutamate come out. These are our vesicles, and we normally have snare proteins here that bind it to our cell membrane. Okay, so we're going to walk a little bit about what it does in the on center. So our on center has another G protein called mGlu-R6. It's a metabotropic glutamate R6 protein. And when it has glutamate bind, it will display a subunit that will close the calcium channel. It's basically a cation channel, but we will think about it as a calcium channel for now. It's TRMP1. It's normally open. However, if we have this in here, it will close. We don't have glutamate, so it's not activated. It will keep it open, and then we will release the glutamate from our vesicles. And because we have glutamate here, we have in our ganglia another receptor that's called AMPA. It's inotropic. So once glutamate binds, it lets positive things in, which will depolarize the cell and send an action potential to the brain and will tell it that there's light. The opposite happens when it's dark. If it's dark, no confirmation, we won't have an alpha subunit here. The PDE won't be activated. We won't block our cGMP. We would have sodium come in. Our sodium would open our calcium channel. Our calcium channel will release glutamate, which will activate our mGluR6, which will send a subunit, which will close the calcium channel, which will make no calcium come in, and so we won't have glutamate here. And because we don't have glutamate, our AMPA won't let positive things in, so we won't have an action potential, and that will tell our brain that it's dark. Okay, those are the two options that could happen when we have an on-center. Now let's see what happens in an off-center. On-center uses mGluR6. Off-center uses AMPA in our bipolar cell. So let's say there's light. 11-cis-retinol transfers into trans-retinol. We have our PDE. It blocks our cGMP. No sodium, no calcium in, nor glutamate. No glutamate, no activation of AMP. So we don't have positive things in, which makes it no action potential, no calcium things in, no glutamate release, no AMPA opening here, no positive things coming in. It won't set an action potential. Okay, that's when it's light. What happens when it's dark? Okay, no change in transformation. We have our cyclic GMP. Let sodium in, calcium comes here, binds into our vesicles that are attached to our center proteins. We do have glutamate. That glutamate attaches to our AMPA. Our AMPA lets positive things in. That positive things in opens our calcium channels. We will send glutamate and we will bind to our AMPA, and now we will let things in. And because we let things in, we have an action potential, which will tell our brain there's light. So they basically work backwards. Our on-center likes light, our off-center doesn't like light. So off-center activates when there's no light, on-center activates when there's light. Let's talk about this a little bit more. So we know that we have our photoreceptors and our bipolar. There's something in between that's called a horizontal cell. So we'll think about this as an off-surround, on-center. So let's say we have light in our off-surround, and when there's light, we already talked that there's no glutamate. Glutamate normally will make our horizontal cell release GABA, which inhibits the on-center from releasing glutamate. Okay, we have less glutamate here, less GABA, less inhibition, more glutamate coming in. Because we have more glutamate coming in, that will activate our mGluR6, which will close this channel, and we won't have an action potential in here. On center, what happens? We have light, we won't release glutamate, we don't activate the mGluR6, therefore we leave our channel open, which will create an action potential. So we count this as an action potential. These two work exactly the same, so it's a no from here. So now we count. For this purpose, we'll count our off surrounds as having 2 action potentials and our on center as producing 6 action potentials. So since we don't count this ones, we represent this as having 6 action potentials. 1, 2, 3, 4, 5, 6. Let's do another example. This time, off center, on surround. We have light. If we have light, we have less glutamate. Less glutamate will give us less GABA, which will inhibit glutamate less. And because we have more glutamate, our AMPA channel will let positive things in. Therefore, we will have an action potential, so we will count this. Real quick, something that I forgot to mention is that when we have our off center, we use an AMPA receptor. When we have on center, we use mGluR6. That's why they do things differently. Let's go here again. Off center, we have light, no glutamate release, no glutamate release, no AMPA activation, no AMPA activation, don't let positive things in. Therefore, we don't have an action potential, so we don't count this one. Our on surrounds are the same, so if this is a yes as well. So 2 plus 2 equals 4 action potentials to represent this. 1, 2, 3, 4. We also see them as these circles, as positive, negative. Positive means on, negative means off. So in this case, we have a positive center, an on center, off surround. So for this example, I decided to just make this thing dark, and then we will have light in everything else. So we said that it's 2 for the surround, for each surround, and 6 for the center. Same thing here. Okay. It's off surround and it's dark. It would be activated by, like, we would have an action potential. It's an on center and it's light. We'll also get action potentials from there. It's an off surround, but it has light. We don't count it. So 6 plus 2 equals 8 action potentials. So we represented that here. Now we're doing exactly the opposite. We have an off surround, on center. And we decided to make the shade in the same exact part so that we can contrast what it does. So it's an off surround, but it's dark here, so we don't count it. It's an off surround, it's an off center, and it's light, so we don't count it. It's an on surround and it's light, so we do count it. That's why in this case we have 2 action potentials. Thanks for watching.